right, I think we're fairly stable, so I'll kick off. Thanks everyone for joining this Spatial Conversations webinar. I'm Brett, the Market Development Manager for ANZ from Spatial from 10x Genomics.
And I'm Marta Gabrielska from Millennium Science, the Next Genomics Account Manager.
Now, for those of you who are new to the series, the idea is to get more of the background information, unpublished details about performing spatial experiments into the public domain. We hope you find the series valuable, and we encourage you to ask as many questions of our speaker as you have, because he's definitely ready for the hard questions today on all your bioinformatics queries, or at least some of them related to the talk. He's obviously put a lot of work, a lot of time into thinking about these experiments, and we are really sure that you're going to hopefully learn a lot from today that you can't normally access outside of a one-on-one conversation.
Yeah, so it is my pleasure to introduce our speaker today, Associate Professor Davis McCarthy. He's the head of Bioinformatics and Cellular Genomics at St. Vincent Institute of Medical Research. And Davis, the stage is yours.
Thank you very much, and thanks a lot for the invitation to share our work and thoughts today. And yeah, look forward to some good conversations. I think that's part of the appeal of this format is I won't be monologuing for too long. I'll give an overview of our work, and then we can have a discussion. I certainly look forward to lots of thought-provoking and challenging questions. So with that, I'll tell you about this lung disease project that I've been working on with my team and colleagues in the US for a couple of years now. This is a big interstitial lung disease project and collaboration that involves my team at St. Vincent's Institute of Medical Research in Melbourne, Nick Banovich's team at TGen, the Translational Genomics Institute in Phoenix, Arizona, and John Kropsky's team at Vanderbilt University Medical Center in Nashville, Tennessee. So we're broadly interested in kind of many aspects of interstitial lung disease and trying to do a better job of understanding and ultimately treating that set of diseases. And given our own interests, we're very interested in combining genotype data, single cell RNA-seq and single cell omics data, now spatial omics data, and other sort of epigenetic or other omics data like ATAC-seq together to fill out the picture of what's going on molecularly in these diseases, to identify disease cell types, discover more about the role of genetics in these diseases, understand mechanisms, and especially close to my interests and my group's interests, develop bioinformatic and computational tools to do a better job of analyses in these really interesting but also challenging projects that involve lots of different data types and also new technologies like we'll talk about today. Within the big picture of interstitial lung diseases, the focus for our work is on pulmonary fibrosis and particularly idiopathic pulmonary fibrosis, which, as I embarrassingly recently learned, idiopathic is just kind of the medical name for we don't know what causes it. In these pulmonary fibrotic diseases, we go from the health and back and forth as needed when we're breathing. When we have fibrotic lung diseases, we find that the shapes of the alveoli change, their walls thicken, the spaces between the blood vessels, capillaries, and the alveoli fill with fibrosis, essentially like scar tissue. And over time, these diminish lung function. And generally speaking, these are progressive diseases, which is really tragic because once you're on the path towards fibrosis, we don't currently have any good ways to reverse what's going on, to revert back to healthy lung tissue. So there are lots of causes of interstitial lung disease. Some are known. Exposures to environmental factors are important. Think of asbestosis or silicosis or exposure to mold. Some are also autoimmune related. For example, sarcoidosis or formation of cysts. As I mentioned at the start, a focus for us, idiopathic forms of pulmonary fibrosis, those that don't have a clearly established cause, but we do know from other research have a substantial genetic component. IPF sadly carries a very poor prognosis. A median untreated life expectancy from diagnosis is 3 to 5 years. There are some drugs that can minimize progression for a certain number of patients, but they don't work for everyone. And for many people, the only option is a lung transplant, which often they don't get in time. So it's a really problematic disease, and it sits in this awkward state. It's not a rare disease, but it's also probably not common enough to be frequently on the radars of most people. Although once I've started working on this disease, it's amazing how many people I've found, sort of second or third degree who've had someone suffer from these lung diseases. There's more and more background information supporting what we can do now, including lung atlases, which John and Nick Banovich especially have contributed to, and single-cell RNA sequencing, including work that we've done to call single-cell expression QTLs in the lung, can inform our knowledge of cell types that are relevant and genetic factors that contribute to IPF. But it's also quite clearly a very spatially heterogeneous disease of the lung, where even in someone with severe disease, they will have areas of healthy alveoli, areas of very severe fibrosis, and areas that are transitioning in some sense between a healthy state and a disease state. And so for John and Nick, thinking about the biology and the clinical needs in this area, adding spatial context to our molecular understanding of this disease built up through single-cell RNA sequencing and GWAS and other techniques that we are all familiar with, adding spatial context seemed really valuable. And that is, of course, what prompted us and them to try to apply the new spatial omics platforms to this challenge, to understand more about the fine molecular details of the progression of idiopathic pulmonary fibrosis with spatial context. So the study I'll talk about today is our sort of really first foray into this, but nevertheless reasonably substantial in that we've got 9 affected donors, 26 pulmonary fibrosis donors, 12 of which have IPF and 13 or 14 have other forms of interstitial lung disease. And from those 35 donors, we have a total of 45 samples, so tissue punch biopsies from these tissue samples, typically between 3 to 5 millimeters in diameter. And we have the samples themselves scored from pathology as either unaffected, less affected by fibrosis, or more affected by fibrosis. And John Kropsky is the clinician in this project who can make those determinations for us. So we're linked up from the bioinformatics to the omics to the clinical side of things here. And so after some thinking and some pilot work, we ended up settling on using the 10x Xenium as the platform for our spatial omics. We used a 343 gene panel for this work. That's a base panel of around 250 lung genes developed by 10x, and then another 90 odd custom or specifically chosen genes by Nick and John with their specific knowledge of the biology of IPF in mind. Across the totality of the data set, that led us to a data set of 300 million transcripts across these 45 individual samples processed in a multiplex way on the Xenium, which as a bioinformatician is both pretty exciting and daunting to have this massive data set when it comes to the number of transcripts detected. And that's relevant to the way that we've gone about some of the analyses as I'll describe today. So from a bioinformatician's point of view, the Xenium data can kind of look pretty simple in that we have these detected mRNAs. They have a gene label, and we get that off the machine with X, Y, and technically Z, although mostly we ignore that, and an XY location for the spatial coordinates of the detected transcript. On top of that, there's also DAPI imaging to help with identification of nuclei and given when we did these experiments, generated this data before the recent addition of advanced chemistries and reagents for helping with cell segmentation. The cell-based analyses we used what existed at the time of the sort of expansion method from 10x for defining cell boundaries from the nuclei. So that leads us to some of the challenges that we face. And for me, there's two big ones that I'll sort of mention when thinking about how we do these analyses and what we were seeking to overcome or work around or work with when it came to some of the analyses we did. So we want to think about the cell as the atom of biology, and we'd love to get perfect summaries of data at that individual single cell level. That's not exactly what we get off the instrument. What we initially first get or fundamentally get are the observed transcripts with their location and their label. And then there's a task for assigning those transcripts to cells or nuclei. And so we'd love to have that picture in the middle where the cell boundaries are very well defined, and we can very accurately make a call about whether a particular transcript belongs to this cell or that cell or no cell. But as I said, the segmentation as it stands is not perfect, and that leads to transcript assignment with some degree of error. And that means both transcripts that perhaps should be assigned to a cell that are not, and also transcripts that are assigned to the wrong cell or improperly assigned to a cell at all. For me, there are a couple of considerations when thinking about the challenge of cell segmentation and transcript assignment. Some can be solved, like better staining of the cytoplasm, cell boundaries and things like that, cell membranes and so on, can be part of the tractable part of that. We can do a better job of defining what we observe as the boundaries of cell, and that will immediately help with transcript assignment. But in these tissues where we take a small slice and put them on the slide and record XY coordinates for observed transcripts with a sort of a minimal Z dimension, there's also the challenge of just having cells that overlap each other. And so when we look from top down, down the Z-axis into the XY plane, then we might have partial areas of cells that overlap with better defined cells. And so we have transcripts from two different cells that end up being assigned to a particular cell that we observe with some level of confidence. And that's the sort of the more intractable part of cell segmentation that I think doesn't have such an easy solution, perhaps technologically. So that's a whole set of challenges that we might keep in the back of our minds while we try to do good data analyses when it comes to these things, and also things we might think about in the process of developing new, better bioinformatic tools for helping with these sorts of analyses. The second big challenge that I'll mention is this fundamental challenge that I think exists everywhere in omics, but is particularly salient in the spatial omics. It's the challenge of how do we compare highly heterogeneous samples. So as I mentioned, like the motivation for doing spatial in the first place, especially in IPF, is that we've got lungs that have really different regions with different molecular states and morphology and things going on. And yet we need to compare them across multiple individuals so that we're finding the sort of robust and generalizable conclusions that we want out of these sorts of studies. So there's a deep question there about how do we take these very highly heterogeneous samples where what we observe is dependent on the region you happen to have chosen to sample from your tissue, and tissues that are even within the area that you have chosen to assay are very highly heterogeneous. How do we make them comparable to other samples with and without disease and so on? So one question we might ask ourselves are, what are the shared structures and features between samples? So what are those things that are shared in some sensible way that we can then compare between people or between samples? And from a sort of computational perspective or data analysis perspective, we might frame that as, can we find new spaces or representations of spatial transcriptomic data that enable these robust comparisons between samples that we want to make? And again, part of the bioinformatics approaches that I'll describe in passing in this talk today are about finding those new representations of new spaces that make robust comparisons available to us. So that's some of the background thinking for when we go back to this particular data set of these 45 particular samples that we multiplexed, as I said, on the Xenium, and how we thought about things we would need to tackle in trying to do a first good, robust analysis of this big data set. In our case, we had sort of 3 workflows operating in parallel here. So the samples themselves were taken from the biological tissue samples and put into tissue microarrays such that we could multiplex multiple samples, 8 to 12, on an individual Xenium slide, which has the advantages, of course, of reducing the cost of processing per sample and also potentially adding a level of robustness because we have those individual slides or batches with multiple samples such that we can look at effects across slides if we need to. On those slides, we have the H&E imaging as well as the Xenium data. And so that enabled John Kropski and his team to do the histopathology annotations on these samples and identify in kind of the standard pathology way different regions, fibrotic, healthy, airways, other important features of what was going on. On the sort of spatial omics side, of course, we have the Xenium data, and Nick's team, led by Annika Vannon, undertook a cell-based analysis approach. So doing, well, I might think of the standard thing, we assign the transcripts to cells or nuclei as well as we can, and then apply workflows similar to standard single-cell RNA-seq workflows, and then map the cell types, the niches, as we call them, that incorporate multiple cell types together back to their spatial locations and understand them that way. And then the approach that was taken in my team, led by Ruqian Liu, was to work directly from the transcript level data themselves, the individual observed molecules, to bypass the cell segmentation and develop transcript-based niches with a particular approach that I'll mention in a minute, that lets us, in a complementary way, do all sorts of deep analyses without having to worry about whether cell segmentation is working well, introducing errors or otherwise. And so for us, these 2 parallel approaches, cell-based and cell-agnostic, were useful to do side by side. And when they converged on the same sort of answer, that gave us a lot of encouragement that we were in the right direction. I'll finish by describing an airspace analysis, which is another way of sort of defining structures, in this case, airspaces that are naturally comparable between different samples, even between the healthy and diseased. So as I mentioned, from the spatial omics data, the Xenium data, we had these 2 workflows, the one cell segmentation based, undertaken in Nick Banovich's team on the top, and the cell-free based one, the transcript based one on the bottom here, which uses graph neural network methods as the basis for finding a new representation of these transcripts that summarizes local neighborhood information for each transcript that lets us build up a molecular picture of these transcript-based niches. So what is a graph neural network? I might save that to question time, but I'll tell you about this method we use called GraphSage, which is a type of graph neural network, which takes the observed mRNA molecules and their spatial coordinates and first builds a graph. The abstract sort of mathematical notion of a graph. We have nodes and edges in a graph. In this case, the nodes are the mRNA molecules, the transcripts that we observed, and we draw an edge between 2 nodes or between transcripts if they're close enough in space within a defined radius, which in our case is typically between 2 and 4 microns. So if they're within that distance, we connect those 2 transcripts together in the graph by an edge. And doing this, we can build out a graph, a spatial graph of all of the mRNAs observed. those spatial graphs are then run through GraphSage. The SAGE part of GraphSage stands for sampling and aggregating. So what the GraphSage method does is use neural networks to learn functions that essentially aggregate or summarize the material from the local neighborhood around each transcript, but with a degree of sampling that both reduces the computational complexity and adds a degree of robustness to how we incorporate information from each neighborhood around each transcript. What that then yields is what we call an embedding. So a new representation of each transcript in a new space called an embedding space that incorporates that information not only of the transcript itself and what gene it came from, but what the nature of its local neighborhood was like as well. So when we put that method inside a larger analysis workflow, what we do is take that Xenium data, those observed transcripts with their gene labels and XY coordinates, we build the graph as I described, and then we sample small subgraphs, as they're called, from that very large graph from each sample. And we take many subgraphs, so tens of thousands from each individual sample, and in a big joint effort, put subgraphs from all the 45 different samples that we want to analyze together into the training procedure for GraphSage. And so that's important because we're training a GraphSage model on all of the whole data set, which we think adds a lot of robustness and avoids some of the traps of batch effects and so on that we might otherwise suffer from in this sort of analysis with lots of different samples, lots of different batches, lots of different slides. What we get is then a new embedding space, so a 50-dimensional representation of each transcript. And you can think of that kind of like a complicated way of doing principal component analysis where you might take your single-cell RNA-seq data, do PCA to get 50 principal components that provide a new representation of the single-cell RNA-seq data. We're doing a similar thing in defining that new embedding space. We're just going through this graph neural network method, GraphSage, instead of something more appropriate for other data sources like PCA or something like that. With this new embedding, we've got a new space that actually has all of the different samples merged together. So that's a really nice basis for starting to do sensible comparisons between different samples from different individuals, from different tissues and so on. In the embedding space, we can do clustering to do hard clustering, define clusters of similar transcripts in that embedding space, meaning that those transcripts have similar local neighborhoods. And then, of course, we can map those clusters or those niches, as we call them, back into the spatial coordinates in the tissues to see where they fall. And of course, happily, what we see is that there seems to be a lot of biological information in those clusters or niches derived from the GraphSage embedding space for these transcripts. When we do this, and here these little pictures are flicking through just different clustering resolution in the GraphSage embedding space, but we can see both a lot of spatial structure immediately evident and also some really massive differences in what we see between the unaffected on the right and the more fibrotic samples on the left, which are both much denser, filled with fibrotic tissue compared to the unaffected, much more spacious, as they should be, with many more healthy alveoli present there that are open and operating effectively as they should in the lung. When we then look at these, just the proportion of transcripts from these the 12 different niches that we define, and look at them across the different samples ordered from most healthy or unaffected on the left to most pathologic on the far right, you can see, again, very drastic differences in the proportions of transcripts that belong to the different transcript niches as defined by GraphSage. And working closely with Nick Banovich's team and John Kropsky's team, we can assign sort of biologically relevant identities to these different niches. And so very obvious is that you have the healthy alveolar niche T4, which is very highly represented in the unaffected samples towards the left, and that diminishes greatly as we go to more pathologically severe samples on the right. And then we have fibrotic niches T6 and T9, so the lilac and the brown, that are very low in proportion in the healthier samples, and obviously increase drastically there. Also, even things like immune niches, so the sort of almost turquoise and black, that are fairly low in the healthy samples as well, but increase greatly in the fibrotic samples. So we can see these drastic differences in the nature of the transcripts that are present in these different samples as we go from unaffected to less fibrotic and to more fibrotic. Going even sort of more specifically, we can actually associate some interesting pathological features that are present in the H&E, like this phenomenon of epithelial detachment, with specific things going on in the transcript molecular data. So this epithelial detachment phenomenon here actually associates really strongly with this T3 niche, which we call transitional epithelial, which butts up right against the severe fibrosis there. And that's sort of zooming in to show that transitional epithelial state really matching up well with the histopathology observed epithelial detachment. So that, as I said, the graph stage approach gives us a new embedding space, a new representation of these transcripts that lets us make comparisons in hopefully a sensible way between these very structurally and molecularly different samples at large scale. We might think about other ways that we can find, those comparable structures that allow us to make robust comparisons between very different samples. And so the other way we approach that, or the next step for us, was to look directly at the air spaces, which of course are relevant for the lung. And so using some more machine learning approaches, John Cropsey's team identified the air spaces, and then we characterized each of the observed air spaces by the cells and transcripts that lined, or by the transcripts in the cells that lined the airspace. And so again, we had sort of a new way to define the molecular state of these particular structures that we could then compare across the different samples. And so across all the samples, as I said, we identified all of those air spaces, and then we ranked them by the proportion of transcripts that were in that T4 niche. So that was the healthy alveolar niche. So again, now instead of having a ranking sort of from healthy to diseased at the sample level, we can have a ranking of sort of healthy to disease at the individual airspace level. And then we can take a deeper look at what's going on with gene expression across that sort of pseudo time of disease at the airspace level. And so what we find are basically three major classes of genes that change in their expression from quote unquote healthy to severely fibrotic or end stage disease. And so they're essentially genes that are highly expressed in the unaffected healthy state and lowly expressed later on. They're what we call early transition genes that peak in the middle of this trajectory. And then the late remodeling genes, those genes that are highly expressed in the late stage disease, the heavily fibrotic areas. And to do this accurately, you have to take account of the cell type composition of the different air spaces, which Ruchi and Liu did with just some smart application of spline-based linear models for doing this. What was interesting as an overall high-level outcome of this analysis was that the majority of these early transition genes, which have altered expression along the disease progression, were found in epithelial cell types. And this leads us to a sort of a current thinking about the challenge of treating IPF is that if you're already in the late remodeled state, so the very fibrotic state, it's very hard to imagine what you could ever do to turn those very fibrotic regions back into healthy alveoli. But what we're seeing with the early transition gene expression signature are regions that morphologically look okay still, but are molecularly already on the path to fibrosis. And so John and Nick's current thinking is, well, that seems to be the most promising area where you might be able to intervene to prevent further progression of the disease. So that's kind of where we're thinking there. So to summarize, we applied these cell-based and cell-free GNNs. I didn't talk about the cell-based stuff in any detail. That's in the paper. And we liked the complementary approaches of doing it two ways. We identified these cellular interactions and association with histopathological features. We saw that structurally preserved regions in that early transitional state have actually undergone already molecular reprogramming that puts them on the path to fibrosis. And the totality of these analyses leads to indicating that early fibrosis starts with epithelial dysregulation before there are structural changes observed. And Ruchen's been working hard recently on actually making the GraphSage approach that we used here easily applicable by others. So she's put together workflows and tutorials that are available at that link, and we can share that again for anyone who wants to follow up and do those things themselves. So that's the paper and the team, and I'll stop talking and hopefully let you ask lots of questions. Thank you.
Thank you, Davis.
Thanks, Davis.
Yeah, it was amazing, I think, and very important, especially the type of questions you're asking. Just a quick housekeeping information. I know we are running a bit over time, but please don't drop. You're still very welcome to ask questions. These are spatial conversations, so please join the conversation. This session will be available on Millennium Science YouTube channel if you need to drop off. And so please, any questions, put them in the chat or raise your hand and just ask the question directly. I think we might start. Brett, I'm sure you have questions.
Yeah, I have one. I can't get past segmentation. We get asked about it so often. There's been a lot of discussion about transcript density-based approaches to segmentation, things like ProSeg and Ficture. Can I ask what your opinion is of these?
Yeah, I think they have merit. So I think my current thinking at a high level is that no one approach perfectly solves any of all of the challenges of cell segmentation. I think ProSeg and things like that think about density are useful. And again, like I think sort of the chemistry-based approaches to doing a better job of cell segmentation will help with the tractable parts of cell segmentation, where there's sort of a clear cell that's nicely, say, sampled in our XYZ slice. So I think they will contribute there and help there. They won't, as I can see, help with sort of the intractable parts of cell segmentation, where we've got the challenges of transcripts assigned to the quote-unquote wrong cell because we've just had overlapping cells in tissue, and some are identified through whatever segmentation method, and some are just not. So I think they'll be part of the picture. I don't think there are any magic bullets, so I don't think they're that, but we are certainly going to be looking at them seriously for our future analyses of these kind, because I think the more tools we have in the toolkit, the better. More specifically, we have tried Ficture. That does some similar things to what we might do with GraphSage, in the sense that it operates at the single transcript level, and it is sort of more akin to factor analysis type things. So a bit of a different approach computationally compared to GraphSage, but can achieve some of the same things of finding those sort of transcript-based niches in the same way as we have done with GraphSage. So that's worked well in our hands and Nick's team as well. So that's also Ficture, particularly, also a good part of the toolkit for sure.
Okay,
Great.
cool. Thanks, Davis. Yeah.
Sorry, I just say, I know our team is looking at deconvolution methods to try and understand the potentials for cells on top of each other. So we're making progress. So hopefully that's something we can report in the future.
Yeah, we're thinking hard about that as well. So Ruchen specifically, who worked through these GraphSage analyses, we're also thinking about ways in which you can use, like local information from GraphSage and other maybe also a priori information to think about what transcripts are likely to be wrongly assigned. If you've got a classic T cell marker in an AT2 cell, alveolar cell, that suggests you might have some sort of cryptic overlap that's undetected, and that might be a candidate for filtering out of that that AT2 cell, or vice versa. So definitely lots of interesting work to do there that I think obviously great that you're working on it. Very excited to be working on it. Move on.
We have some questions for you. And Dina Kazemi has raised her hand. Dina, I just hopefully unmuted you.
Yep.
Please ask your question.
Thanks for the great talk. I have two questions. I think the first one is I'm curious to know why you decided to use GraphSage instead of other GNN architectures like graph attention networks or graph convolutional networks. And the second question is, I think one important hyperparameter in GraphSage is the number of neighbors that you include in the analysis. What sort of methodology or workflow did you use to, narrow it down to the best number, basically? Thank you.
Yeah, two great questions. So GraphSage, I forget the reference off the top of my head, but one of the very early kind of papers used an approach that was not really sort of developed as a bioinformatics tool, but mentioned that it used GraphSage internally. So Ruchen read that paper and thought, okay, well, if it's been effective in that setting, then it's worth a try for us. So that was how we sort of found GraphSage, and we tried it out and found it broadly useful. Where sort of, yeah, I mean, Ruchen's aware of graph attention and other approaches like you mentioned. We haven't done a deep comparison, again, with the sort of approach to this project that our aim was the first kind of sensible analysis that we were happy with, not making the definitive judgment about which method, approach, or so on was the best for all things. So I think, I mean, it's of interest to us is following up on whether graph attention and graph convolutional neural net something could do better than GraphSage. It's interesting. I mean, as I mentioned in passing, GraphSage has the sampling and aggregation attributes that do make it quite scalable. Is it more or less scalable than others? Not yet sure, but good things to follow up. Second part of your question was about hyperparameters like number of neighbors to sample and so on, what radius to choose in spatial distance to build the graph in the first place. I think in terms of radius, there is some sort of biological knowledge that guides that. So if your radius for joining different transcripts together in the graph is on the order of 2, 3, or 4 microns, then you're kind of gathering information mostly at a subcellular level. If you extend that to 10 or 15 microns, then you're sort of starting to aggregate information across them. We did some investigation, some trial and error sort of approaches to decide on the hyperparameters that we used. Like a lot of these, broadly speaking, analysis approaches everywhere, in omics, there are a lot of user degrees of freedom. There are a lot of points in analysis where you need to make a choice and apply your best judgment and do sort of what investigations you can to decide on what you do. So that's essentially what we did here. I expect those things should change depending on the biological context, and what works best for lung might not work best for other things. And then there are trade-offs also. The larger your radius, the denser your graph, the more computational complexity in running GraphSage or similar things. So there does end up being a practical limit. What Ruchen has found that actually recursively applying GraphSage, so doing it sort of a very local summary and then applying GraphSage again at a higher level can be quite effective for finding niches or domains at varying scales.
Well, we have another question from Klaus Leonard. He said, thanks for the talk. Can you comment on scalability of GraphSage with larger gene transcript numbers, for example, Xenium HD?
Yeah, another great question. So I would say it scales very well in terms of sort of transcript numbers, in the sense that the number of observations. We had 300 million in this, and so GraphSage is what they call inductive. So you can train it on a subset of your data and then apply it to the whole data set. So that's really, really good for scalability. What we don't know yet is exactly how well it works when you go from the 300, 500 gene panel to the whole transcriptome, 20,000 or so, if you're getting that out of Xenium HD. We just don't have the data yet to have tested that. and a similar question, Xenium 5K, Xenium Prime, the 5K panel, is it going to work exactly as well? We don't know. We need to test it. I can think of reasons why it is more challenging for a method like GraphSage to integrate information from 5,000 or 10,000 or 20,000 genes in a panel than from 500. So an open question at this stage. From the computational scalability, I'm reasonably comfortable saying that I think it'll be fine. I think it's more of the ways of integrating the information across that panel are as effective when you've got many more, perhaps like non-specific genes for the particular biology or cell types than you have when you have a very, very well-chosen set of 343 genes for lung biology, as we did in this particular case. Having said that, we've seen GraphSage with colleagues at WeHi work really well in different settings, from brain cancer to other things quite different from the lung, but that's still been in the targeted panel yet. So if you've got Visium HD data in large volume, then sure, let's have a go.
And I think this will be our last question again from Klaus. Would it be worth limiting it to highly variable genes?
Possibly, yeah. I mean, I think like in lots of analyses, if there are genes that are not particularly informative, like not statistically informative in that sense, then yeah, you may as well filter them out of the analysis. And I think that I suspect will apply here. So yeah, again, if you've got your 5K panel and 1,000 of them are really lowly expressed, rarely detected, then you may as well filter them out. I don't know if that's the case. I haven't seen enough 5K data to state that as any more than a hypothesis, but yeah.
Well, hopefully we'll see some more soon. And I know
I certainly hope so.
There's certainly some nice new public 5K data sets on ovarian and cervical cancer for those who are interested in checking out our website. All right, look, I appreciate that we've gone a bit over time. Many people have stayed. We thank you for that and hope you found it interesting. A recording of the webinar will be available on the Millennium website. And if you do have any questions, feel free to get in touch with the people from the call. And we have a
I noticed one just from Alex there. He specifically asks, can you comment on general areas of collaboration that would interest you? We're very open to collaboration. Obviously, we're a limited number of people with limited capacity, but always happy to talk. I think things that particularly grab our interest are those sort of projects like this one that I've described, where there's that intersection of really compelling need for study, really interesting biology, and some element of kind of new informatics or computational need. If it's, stuff applying solid techniques that exist already, I think there are plenty of people that can help with that. If you really think there's something new that hasn't been solved, or you've picked up on things I've mentioned in this talk, and like, actually, that particular thing is really relevant, and I've got a good idea or a good data set for testing out or thinking about new methodological approaches, that's always going to excite us. So definitely don't be shy getting in touch. Can't do every possible collaboration out there, of course, but always happy to chat and sort of build the community where we can.
Great. Well, I hope that made it worthwhile for some people to stay a bit longer. Thank you very much, everyone. And we look forward to participation at our next one, which is a talk from Lip and Lou on spinal transcriptomics. Thank you very much, everyone. Bye.
Thanks all.
Bye.